Hello and welcome everyone to NephCure's 20th Anniversary Progress Panel. We are so excited to be with you here today, and we have a great group of panelists to share all about NephCure's progress over the past 20 years and that progress in protein-spilling kidney diseases as well. Just a few housekeeping items before we get started. Attendees, you will be muted for the remainder of the webinar, but feel free to ask questions by using the Q&A tab on your Zoom toolbar, which is either at the bottom of your screen or sometimes it's at the top of your screen. And as we go, you're able to ask questions, and hopefully some of our medical professionals can answer those. We hopefully will also have time at the end of the webinar to answer a few questions. I want to first start off by thanking our sponsors, Alnilam, Kaneka, Mallinckrodt, and Retrophin for making our virtual programs possible. Next, it is my pleasure to introduce to you Melanie Stewart, who is an FSGS patient and daughter of one of NephCure's founding members. She will discuss NephCure's progress from her perspective from the beginning. Take it away, Melanie.
Hi, everyone. I think you can see me. I don't know what's on. I don't know what you can see, but I can't see myself. But anyway, my name is Melanie. I was diagnosed in 1995 with nephrotic syndrome, and that was right before my 8th birthday. That winter before, I had been waking up puffy, and my mom had taken me into the doctor, and they did a dipstick for protein, and it came back obviously very high and not right. And then I was treated with your typical steroids and immunosuppressant drugs. This was in 1995. It was prednisone, and I'm not sure the immunosuppressant drug, but just your most basic treatments. And unfortunately, then there just wasn't anything else besides prednisone and immunosuppressant drugs. I did a course of IV prednisone, and I was just very, very puffy. There was just, I never had any sort of like change in the nephrotic syndrome or FSGS. And within about a year and a half, I had progressed to end-stage renal failure. And in 4th grade, I started peritoneal dialysis. Maybe if I was going to say there was a highlight in all of all the problems that I've had after I started peritoneal dialysis, I was actually a Make-A-Wish participant, and my family and we went to Hawaii, and my wish was to swim with dolphins. So that's what I did, and I think that was just an amazing experience. So like I said, I don't know if there's a highlight to any of this. That was an amazing trip. And then so in 1999, I had a kidney transplant. My dad donated his kidney to me, and like Kelly said, my dad was one of the founding members of NephCure. I think seeing what had happened to me, and maybe even more so happening to his own kidney, was maybe a reason, if he didn't have enough of a reason already, but definitely probably a driving force to like start NephCure, or find something to do differently. As soon as I was transplanted, the FSGS reoccurred immediately. Just never didn't. It went straight back to FSGS. And I had so many complications and challenges with the transplant. I had post-transplant lymphoma. I became septic from the plasmapheresis catheter. It was, those 2 years, unfortunately, were just the worst. And then they ended up removing the kidney just less than 2 years from when the kidney was put in. And so I think I had written this down that week that I had my transplant removed. I was due to go to Washington, D.C. and testify on behalf of NephCure. It was one of the first, not events, but one of the first events that NephCure had really participated in after its founding. And I wasn't able to go because I had the kidney removed. And so my dad went in my place and gave my speech. And it was just, I remember sitting in the hospital thinking, this is so strange that I'm losing technically a third kidney to this disease. And my dad is down in Washington, D.C. Trying to, get more funding and just more just information, just so people would know about it. And yeah, that was just a very weird time. So after the kidney was removed, I was back onto dialysis. I was on peritoneal dialysis for about another 6 years. I started hemodialysis, and that's where I've been ever since. And I think if my math is right, I've been on dialysis for almost a total of 21 years, which is crazy to even think about, that this is what I've been doing for, obviously, well, I don't even know, most of my life. And certainly over these many years, I've had a lot of ups and downs. The most major events were rare complications from these long-term peritoneal dialysis. And sorry, I just lost my train of thought. I'm so sorry. Where I was kind of going with that, I feel like I've lived a normal life somehow, and I think it's just how I go about life, is thinking this is normal. But I've always had a very positive attitude. I can't stress enough, I don't know how anyone would do this without a positive attitude, and obviously it's hard to do that, but just having a positive attitude has just always been my go-to, I guess. And then, so I wanted to talk a little bit more about how NephCure was founded. It was founded by my dad and another dad that was at the Children's Hospital of Philadelphia. And then there was another two dads that were in Michigan. And through the doctors, they were kind of put together because they were kind of trying to do the same thing. And that's how NephCure was started. And it was started basically in my dad's office above the garage. And it's amazing to see how far we've come in 20 years. Like I said, when I was first diagnosed, the treatments were there just was no options. And I see that there are so many different drugs now that, are very promising, and that all has to do with NephCure and what they've done over these last 20 years. So I wanted to talk about maybe how FSGS for me has affected me. I honestly just don't know anything different besides this. This is my normal. And when I sometimes try to think about my my story, I forget major 6-week hospitalizations, and that's so not normal to forget about that. It's oh yeah, I forgot about that and that. It's just crazy to think about all of the things that I've been through. But yeah, and so NephCure has been such a big part of my life, and I think just knowing now that people have a place to go to get information, to be a part of a community is just amazing. I know when I was diagnosed, my parents had absolutely nowhere to go to find information, nobody to turn to, and especially with the internet now, it's amazing to see the type of community NephCure has created. And I think that's, if I was going to say the biggest impact, is just creating that type of community and being able to share stories with one another. Obviously research is huge, but I think personally having just a group of people that understand also, and just being able to listen and understand is just amazing. I just, little anecdote, my dad always said, the day that NephCure is no longer needed is going to be the happiest day. And I think that is true, but just going back to exactly what I was just saying about the community, is NephCure, even if and when we do find the cure for these diseases, and that part of NephCure isn't necessarily needed anymore, this community will always be there, and that's just going to be a lasting legacy for hopefully a very long time. And then I get, maybe just touching on where I am today. I've been very reluctant to have a second transplant just because of how awful the first one was. And unfortunately for me personally, and this is just my view and just knowing my body and how this disease progressed for me, I just don't see a second transplant working for me. There's certainly been advances in treatments and different medications, treatments, plasmapheresis, the liposorber, and different things like that. But I don't think in my case a transplant would work. So that's where I'm at right now. I'm just on dialysis. I was there this morning. I'll be there Friday. I'll be there Monday. I'm here at my house. My dog is sitting here. Like I said, I think I lead a pretty normal life. I live here with my boyfriend and my dog, and I travel to see my mom and dad. And yeah, I guess that's where I'm at right now. I don't know. I think that's it. I'm just very proud of NephCure, and I'm very happy to be a part of it, and to be able to share my story once in a while.
Thank you, Melanie. Yes, thank you so much. We appreciate you being real and honest, and that's what it's all about. So we appreciate you. Thank you.
Thank you.
All right, so next up is Dr. Alessia Fornoni, an adult nephrologist, who's also the chief of the Kant's Family Division of Nephrology and Hypertension, as well as the director of Peggy and Harold Katz Drug Discovery Center. And this is both at the University of Miami, and she's also a member of NephCure's Scientific Advisory Board. Dr. Fornoni's career has been dedicated to her interests of podocyte metabolism and glomerular disease development and progression. And so I will leave you to take it away. Dr. Fornoni, thank you.
Thank you, Kelly. I want to congratulate NephCure Kin International for the anniversary. Is a true milestone. Thank you, Melanie, and your father for what you've done. We are here today because of people with the vision and the positive attitude as the one you have. So thank you again. I was very touched by your story, and believe it, that's really what brings physician scientists like me really from the bedside to the bench. The title here, from bench to bedside, but frankly, it's really interacting with patients like you that really give us a sense of responsibility and the energy to go to the bench and find what's there better to do for patients. So I'm a true believer actually, and I think the highlight of this presentation today really showed this, that it's only through a true team effort with partnership between academia, pharmaceutical company, biotech firm, foundation, patients, their family, and the regulatory agency is the only way that we can really change the nature of biomedical research and exercise this wonderful mechanism of bringing bedside observation to the bench and back to the patient so we can actually close the circle. So when I'm sure that many of you have heard your doctor saying, I'm sorry, I don't know what causes your disease. And I can tell you, we call this word idiopathic. The fact that we don't know exactly what caused the disease, we tend to say the disease you're having, the spillage of the protein, is often idiopathic. We don't know what causes it. And as long as we don't understand what causes it, we will not know how to reverse it. That's why it's been so important what you have mentioned, Melanie, to actually have so much investment into research that will help us to identify target that will eventually be ideal for drug discovery, so we can bring back these wonderful cells that we all like, the podocyte that doesn't really work in disease conditions, and cure the disease. And I think that's what really brings us here today. But as Melanie had already mentioned, at the beginning, when NephCure was funded, there was really nothing that we could offer to our patient, but thanks to the attention to this proteinuria kidney disease, many targets have been developed in the past 20 years, and it's an exciting time where a lot of these targets are actually being tested in phase 1, phase 2, phase 3 clinical trials, so almost ready to actually come back to the community as an available treatment for proteinuric patients. And I will talk to you about what NephCure had done for me and how actually I believe that the support from the foundation has really somehow gave the foundation to an idea that translated into patients. So we will talk about these targets, a little bit about rituximab. So I've been one of the first investigators, actually, or clinician treating patients with rituximab with proteinuria. And I can tell you, this was not based on evidence, but this was a team decision between the patients and myself. There's nothing else I can offer. This drug may work in my mice. Can we try it in humans if we have nothing else to do? And so it was a conscious decision to do what I call experimental therapeutics, an attempt to use a drug that was developed for a given purpose for another purpose. And what became very clear to me when we start utilizing this antibody that is meant to destroy B lymphocytes, the cells that produce antibodies, that actually the response to this drug was not always related to the ability to get rid of this B lymphocyte that makes immunoglobulin. And that's when NephCure came and offered a pilot study. I was a junior investigator then, and through that pilot study, I was able to discover that this antibody not only binds and destroys the cells that makes immunoglobulin, but also binds this target on the podocyte, and by doing so, actually protects the podocyte. So while the drug was killing the bad cells, making the antibody, it was also protecting the podocyte to prevent the spillage of protein in the urine. This study, on the next page, we can see on the next slides, we can see was initiated based on retrospective data. What are retrospective data? We look at patient chart. The University of Miami is a very large transplant center. Last year, we were the #1 in volume with more than 500 patients. So we went back in time to see whether patients that received rituximab at time of transplantations with FSGS had less proteinuria after transplant. And through this study that was partially funded by NephCure and by the NIH, we were able to see that those patients that received rituximab experienced less nephrotic range proteinuria in red in the first months post-transplant. However, we actually know that Rituximab is not the response for everybody. I'm pretty sure that many of you have tried and did not respond. So as a scientist, we really need to make sure, number one, that we validate these retrospective data with very nice design prospective data. And there's many ongoing trials to test whether Rituximab may be a solution or not, or a valid treatment. So the question for a scientist like me is, what else is there in the lab? So I will talk to you about another potential target, which is ABCA1. And if you go to the next slide, you can actually see that I've been lucky enough to participate to this consortium of investigators that are really studying patients with protein spillage in the urine to try to see whether their kidney can tell us something about what should be the next drug. So I've done this exercise to work with Neptune, the Nephrotic Syndrome Study Network, that really enroll patients and help us studying the patients in a more detailed manner that you would do just by going to your doctor. So we can actually see what are the pathway and the genes that are modulating the kidney of these patients when they're spilling protein. And one of the things we observe is that there were accumulation of cholesterol and lipid inside these podocytes, which were never thought to even be a cells of lipid metabolism. So we went to the bench, we studied experimental model of FSGS, we found accumulation of lipids in the kidney cortex, suggesting that kidney disease with proteinuria are sort of a form of kidney disease, fatty kidney disease, like we have fatty liver. So what did we do next? We worked to identify a molecule that would actually sequester this cholesterol content from the kidney. And on the next slides, you can actually observe that we use nothing but a sugar that sequesters cholesterol out of the membrane of the cells, named cyclodextrin, to observe a dramatic reduction in the albuminuria, albumin-creatine ratio. That's what you do when you check your dipstick, and improvement of the histological feature. So these drugs, which is now moving forward for phase 2 trials, I hope it's gonna show us some data in the future. But again, as a scientist, I want to know more. I want to know what are the mechanisms. I want to know whether I can find a small molecule. I want to know whether I can find an oral agent that my patients can take every day without going for IV infusion every week. And this leads us to the next slides that says, we didn't give up. We developed a small molecule acting just like the one I've shown you before. This small molecule is well tolerated, already made safety studies in human, and now is ready to start phase 2 clinical trial. That's where we compare the drug to a placebo to see whether the effects in human is similar to the very dramatic effect we have observed in mice with FSGS, where there is a complete normalization of the albumin in the urine in the first graph, of even the serum creatinine, and the mortality, knowing that these mice die actually from kidney failure. So it is an exciting time, as Melanie said, and I want to conclude by saying it's only by, in the next slide, it's only by being here together that we can actually see the ability of the physicians, seeing the patients, bring the patient back to the bench with the help of the NIH, the patient community, the scientists, and then with the regulatory agency and the industry and the patient community, again, bringing back discovery to the bedside. And I want to highlight the importance of patient communities. Patient communities are important both to bring the discovery from their bedside, their experience, we have Melanie today, to the bench, but also to advocate for more attention to go to the disease so that the discovery can translate back into patients. And I want to thank everybody and once more congratulate NephCure Kidney International for this wonderful anniversary achievement.
Thank you, Dr. Fornoni. We appreciate you and all of the years that you have spent dedicated to these rare protein spilling kidney diseases. And we at NephCure love to see the success that you continue to have in your discoveries moving forward. So we really appreciate it. Next, I'd like to move on to introduce Dr. Stuart Shankland from the University of Washington in Seattle. He's going to be speaking about the podocyte. And just a little inside tip, if you ever want to get a bunch of nephrologists passionate about a discussion, just bring up the podocyte. And so I'm looking forward to hearing about Dr. Shankland discuss the podocyte.
Well, thanks, Kelly. The title I was given was The History of the Podocyte. I could talk for 5 hours on the podocyte, but we have 10 minutes. So I want to thank NephCure International for inviting me and for the incredible work that they've done to transform not only the science, but the patient advocacy and the future of, the current and the future of problem. They truly, as an organization, have been game-changing for the past 20 years, as we heard from Melanie's story and from Dr. Fornoni. So what I'm going to briefly talk about in the next 10 minutes is give an overview of the podocyte, a little nerdy conversation about what that is, put that into some context. And then I was asked to talk about history. So I'm going to have 5 examples of game-changing discoveries and events and how we've got to where we are today. And then what really is the future that we can expect. So that's really what I'll be touching on. Can I have the next slide, please? So this is the kidney on the very left. I don't have a pointer, so we're just going to have to listen to me if you don't mind going through this. The kidney on the left, and the middle panel over here drills down a little further. And the little ball is the glomerular. That's really the focus of the scene of the crime. In the top right panel, and moving to the right over there, is a glomerulus looked down from above. Now, obviously, we don't have different colored podocytes in the normal situation, but there's different cells of different colors. Thanks, Kelly. Great job pointing for me. Really highlight some of the podocytes. And then if we go further down to, on the left there, thank you, that's really looking down from above. Those beautiful orange colored cells, for the sake of this picture, are really wrapped around those blood vessels. Next slide, please. So this is a cross section through a glomerulus. And what I want to show you on the bottom slide is a schema of the glomerulus. And there's white holes over there, there's capillary vessels. And on the outside are these yellow cells, the podocytes. And if we look to the top right, cross-section glomerulus, the bottom is the capillary, that's the blood vessel. And we don't want good things like protein slipping through there. So this is what we call the glomerular filtration barrier. And it has 3 different layers. The innermost layer is the endothelial cell. Then you have the glomerular basement membrane. And on the outside are the podocytes. These 3 layers are intact, and one of their major functions is to prevent the escape of protein and other good guys going from the blood space to the non-blood space, which we call the urinary space. And I just want to take you down to the very bottom picture over there, which is a transmission EM, where the arrow is perfectly situated. Just go down a little bit more. The bottom arrow is looking in the blood vessel. That's right in the blood vessel. And if you go up to the top where the black little arrow is, that's the podocyte. And that's really what is injured in the majority of nephrotic diseases. Next slide, please. So on the left panel over here, where I've written control, those are the normal podocytes. You can see they're sitting very nicely on its layer called the basement membrane. And on the bottom picture over there, you can see the podocytes in B, panel B. They're all flattened and horrible looking. And that's what happens in many nephrotic syndromes. And so we've known historically that the podocyte is involved for a number of years. And on the right over there are the diseases that we typically associate with podocyte diseases. And the traditional setting was membranous nephropathy, FSGS, or focal segmental glomerulosclerosis, which we heard from Melanie, that she has that condition, and minimal change. But as the field has evolved, there are a number of other diseases, diabetes and others, that also have podocyte involvement. Next slide, please. So I'm going to show you 5 examples of the history of the podocyte that has got us to where we are today. In 1998, this was a game changer. And so what Dr. Tryggvason found was that in congenital nephrotic syndrome, he and his group, mutations in nephrin. Now, what is nephrin? If you look at these 2 pictures above, and maybe you can just look at my hands, the podocytes are sitting like this on top of one another and preventing protein from getting through over there in these little slits. And what Dr. Tryggvason discovered was that one of the major proteins that prevents protein slipping through here is nephrin. This is a real game changer. Not only did it link a genetic condition with a clinical condition, but it allowed the discovery of numerous proteins that really make up the ultrastructure of the podocyte. And now we know that there are over 40 monogenic diseases, genes that have been discovered, mutations that give rise to nephrotic syndromes. And this is an ongoing discovery. And it has allowed us to really think about treatment approaches, as you heard from Dr. Fornoni. Next slide. Because of this, really, clinical diagnosis really changed the game in many ways, because we can now go back to the bench and use these discoveries to look at specific proteins. And as a result, from many mouse studies, we certainly, many groups across the world can now delete or knock out a specific gene of interest or overexpress it or color it. As you see in the bottom left of here is a picture from our lab where we can actually label podocytes green in this case and see what happens to them. And because of all of this, we've been able to get cells in the cell out of the dish and grow them to see what conditions may affect them and how we can have potential treatments. And as Dr. Fornoni said, this allows us to go from the bench to the bedside and back again. Next slide, please. 2009 was a real game changer for the podocyte world, where Dr. David Salant from Boston found the protein that is responsible for membranous nephropathy. It's something called phospholipase antigen receptor 2, and really changed the game of membranous nephropathy, where now we know what the antibody is. There are different antibodies that can be, the levels can be measured in the blood in terms of how we think about membranous nephropathy, which is a podocyte disorder. Next slide, please. Oh. In 2010, my good buddy Martin Pollack at Harvard really also had a major game-changing discovery in the history of the podocyte. It's been well known that African Americans have an increased incidence of FSGS outcomes compared to other racial groups. And Martin discovered this in very, very important gene called APOL1 as a major risk factor for African Americans and FSGS. And now the field is moving from that history to the current future, to the current, where trying to understand how does APOL1 actually affect the podocyte, and in doing so, can there be particular therapies that we can target? Next slide, please. The final, can I have the next slide, please? Thank you. So in terms of the history of the podocyte, I've given you some game-changing discoveries, but now I want to just finish here with some game-changing events. And this is what we have called the International Podocyte Conference. And the first one was in 1998. I remember going to this in Germany, and we didn't think this would really take off. But after thinking what the success of this, what has happened is now we have up to 13 of these international conferences. And this is where NephCure has really been a game changer for the field, has really supported this very, very important event where international scientists come together. And Unfortunately, it was cancelled in 2020 in Manchester because of COVID. But for the first time, the meeting has evolved a little bit in that patients are also being involved moving forward. And so this has really brought the scientific community together. It is now bringing the patient community into this important top-level science and discovery. And again, NephCure International has a major role in these meetings. And the picture on the right just shows Josh opening the 12th conference in Montreal, Canada. And so the question now is, sorry, can I have the next slide? Oh, there we go. The next slide, please. Thank you. So what does the future hold for the podocyte? I've given you the history. Ultimately, really what we want is precision medicine. And I have 1 more minute left for this talk over here. And so what we really talk about precision medicine is really, we want to have a specific diagnosis. We want planned, targeted treatment for a patient. We want to find out, is the treatment working or not? When do we stop it? When do we add another one? And what prognosis can we have? And what this requires is that we take the person's own genes, their proteins, their molecular makeup, and we put this into the blender. But on the left over here are some important studies that you'll hear about for a moment, likely from Dr. Mariani, talking about Neptune, CureGN, kidney precision medicine. We put these in the blender and we add a variety of genetic studies, biomarkers and antibodies and molecular kidney pathology, all this exciting stuff where the future of the podocyte is. We put these all together and we're able to make a diagnosis and a treatment and really tailor it to the patient. So we've come a long way in the history. And if I can have my final slide, please. And that is, I'm not a big quote person, but, oh, I don't have a quote. So the quote disappeared. I forgot to put it in there. But essentially, history is critical. But what this tells us is what we're missing and how we're moving forward. And with NephCure Kidney International, the field is moving dramatically forward in the podocyte. And thank you so much for your time. I look forward to some of the questions on the chat. Thank you.
Thank you, Dr. Shankland. We appreciate you so much, and we love hearing about the podocyte and really how the history, how it's evolving right before our eyes is very exciting. So next, I'd like to introduce to you Dr. Laura Mariani from the University of Michigan. She's going to be talking about Consortia studies and how these longitudinal studies have become an integral part of the progression of our rare kidney disease. Dr. Mariani is a co-investigator with both Neptune and CureGN, and she also sees patients, adult patients with glomerular diseases, and teaches at the University of Michigan. Thank you, Dr. Mariani.
Well, thank you so much for inviting me to participate, and congratulations to NephCure on this amazing milestone. So I wanted to start out with first on my next slide, an example of kind of how as a clinician, I really fell in love with taking care of patients with glomerular disease. And kind of this, I think, is a good example of why all of us feel that advocacy and research is so important to improve the care we're providing for these patients. So for example, patient A is a young man, initially was born in the Philippines, but then grew up in the United States, completely healthy until around age 20, when he showed up with classic nephrotic syndrome, spilling a ton of protein in his urine and a lot of swelling. And thankfully, when we put him on immunosuppression, the disease went quickly into remission. Occasionally, he would lose his health insurance and wouldn't be able to afford his medications. The disease would come right back. But when he reconnected with care, we'd put him right back on medication. And again, the disease would go into remission. Whereas my other patient, a young woman who also was very healthy, she'd been born prematurely, but had no other real medical problems. Similarly, in her early 20s, she had onset of nephrotic syndrome. But when she showed up to care, already her kidneys had declined in their function. We tried multiple immunosuppressants, none of them worked. And when I completed my training, she was starting dialysis. And so as a clinician, this is incredibly frustrating because the label we give them is FSGS. Their biopsy looks similar, and it doesn't tell us everything we need to know about how they're going to respond to therapy and what's going to happen to them over time. And so to me, this is the real opportunity that we're taking care of patients every day, and we should learn from each and every one of them. So on the next slide, I think that this kind of observations that we see in the clinic are really the fundamental idea behind these glomerular disease consortia. But essentially, as we're taking care of patients with glomerular disease, that we should partner with them and we should learn from them. And that if we could really do that, that if those patients are willing to partner with researchers, we can better understand the underlying biology. And maybe we would be able to come up with a better classification system than just these broad categories based on the biopsy. And if we had better categories, then we would be able to do exactly the type of work that Dr. Shankland and Dr. Fornoni are talking about, which is understanding the underlying biology to have targeted treatments and improve the health outcomes of these patients. So on the next slide, I just want to highlight two of our observational studies. And there are many, many groups, obviously, that are working on trying to better understand glomerular disease. I just wanted to highlight these two, number one, because I've been involved in them, but number two, because NephCure has played such an important role. They've been partners from the beginning, providing financial support, but also, I think, providing that just kind of push to all the researchers to try to really make sure that these resources are being used and accessed by a whole community of researchers. And in fact, I'll never forget one of the very first Neptune meetings I ever went to. NephCure had a patient come to that meeting, and it was, I think she was in middle school, got up and told her story about what FSGS has done to her and her life. And it was so, I think, relevant and poignant, and I'll never forget kind of how motivating that was to the whole room, honestly. And also that a middle schooler could get up in front of a room of like academic nephrologists was incredible. So two studies I wanted to highlight for you are Neptune and CureGN. So Neptune is funded by the NIH. It's part of actually a group of consortia all on studying different rare diseases. So there's rheumatologic diseases, neurologic diseases, a variety of other conditions, but Neptune is part of that. It enrolls patients from 21 sites, began enrolling back in 2010. It recruits both children and adults, primarily targeting patients with nephrotic syndrome who've not had a biopsy, minimal change disease, FSGS, and membranous nephropathy. And this study is unique in that it asks patients to share, if they undergo a kidney biopsy, to actually share an extra core of kidney biopsy tissue so that we can really work on identifying what is the biology going on in the kidney tissue, in addition to using blood and urine to better understand the disease. CureGN also has funding initially from NephCure as well as the NIDDK. This has many more sites, so there are 69 sites across North America and 2 sites in Europe. It began enrolling patients in 2014. This also recruits children and adults all of whom have had a biopsy that either showed minimal change disease, FSGS, membranous, or IgA nephropathy. And CureGN certainly collects all of those biosamples to understand the biology of the disease. But I think another big goal of CureGN is to try to follow patients for long enough to understand essentially who will suffer the complications not only of the disease in terms of loss of kidney function, but other complications like Melanie's story was telling us, for example, cancer, cardiovascular events, things like that. And so those are the two studies, and both studies enroll patients We ask them to come back to have study visits periodically to share not only their clinical information, but also blood and urine, so that we can partner with them and see how they're doing over time. So on the next slide, I want to give you a couple examples of just really amazing data that these consortia aggregate all together. And it's really through the generosity of the participants and the hard work of our study coordinators at all of these sites to be able to collect this rich amount of data. So first, both Neptune and Cure, when patients have a kidney biopsy, their slides are digitized and are populated into a digital repository that can be accessed by researchers from anywhere. We also collect data directly from patients through text messaging to better understand how their disease is changing on a more frequent interval. And then, like I mentioned, we collect blood and urine. In the Neptune cohort, uniquely captured, asks patients to donate an extra core from their kidney biopsy so we can analyze the tissue directly for what genes are turned on and turned off. And so on the next slides, I just want to show you a couple of examples of the type of studies that are being done using this incredible resource. So for example, on the next slide, one thing we've noticed as we take care of patients with glomerular disease is that although we use their kidney biopsy, just go back one, here. So on the kidney biopsy, when we sit down and look at it with our pathologist, not only do we see the features that help us assign an overall diagnosis, we often see many, many other changes in that biopsy. And it's one of, I think, the real values of these consortia to have that digital archive. But this is a really, I think, sort of simple question, but a good one. That sometimes we look at these biopsies and we can see that some of the filters are completely scarred. And what we hadn't known before is that just normal aging or is that the disease? And so this is a study that utilized the Neptune data and just tried to answer that question. How many of your filters should be completely scarred based on your age and your type of biopsy? And so this really practical information is generated just out of that type of data. And there's many other similar projects to this one. On the next slide, as Dr. Shankland mentioned, one of the things we really want to be able to tell our patients is to give them better information about prognosis. We know that this disease is unpredictable and leaves patients wondering things like, is it okay for me to take this trip? Can I take this promotion? Things like that. And so this was a really great study where not only consortia data was used, but also some electronic health record data and some clinical trial data to take a look at essentially when can we measure someone's proteinuria and decide, do we think the therapy they're on is working? And so this is really important. This group did a really great job of looking not only what your proteinuria should be to be informative, but then at what time point, month 4, month 8, month 12, to know is a therapy working or not. And on the next slide, I'll just highlight this that I know a couple other people are going to pick up on. But what we really need are more targeted therapies. And so the benefit of these consortia is that we have this rich data set where we know essentially how patients are being treated, what happens to their proteinuria and their kidney function over time, and we can link that to all this incredible data. So for example, if someone is willing to share their kidney biopsy with us, we can test which genes are being turned on and turned off in the kidney. And we can use that information to group patients and try to better understand for an individual person which biological processes are more active or less active. And so this is data from Neptune where we're trying to do exactly that. On the next slide, I want to show you that kind of these types of studies are really important, not only for giving information to our patients, but also for developing better clinical trials. So for trial design in nephrotic syndrome, it's challenging because these are rare diseases, but there's a lot of variability in treatment response. And it often takes years until patients develop kidney failure. So to know if a novel drug is working, we can't wait that long. And so studies like I mentioned about that proteinuria assessment have really been helpful for trial design. And I think NephCure supporting that particular work has been incredible in terms of making companies feel that, that nephrotic syndrome is something that they can invest in and research and trial their medications. Additionally, we know that patient-reported outcomes are really important. I don't want to just fix someone's proteinuria and make them feel terribly. And so these consortia gather data on how patients feel. And so this is pictures from a study where we're just capturing physical health and mental health. And you can see there's a huge spectrum of how these diseases affect how patients are feeling. So, so patient-reported outcomes, I think in the future are another way that we could help trial development. So on the next slide, I just want to tell you what I hope the future holds. So I hope that this data that our participants have participated get used by as many people as possible, because what we really need is a better understanding of biology. And so we need researchers that study, you know, basic science, clinical science, all of those things to better understand the biology. And on my last slide, I think one of the great strengths is that these consortia bring together a huge number of people. And it's really fun for all of us to come together, patients, bioinformaticians, clinical scientists, epidemiologists, people that study mouse models, geneticists, all of those people, to get them excited about changing the way that we treat nephrotic syndrome. And I think together we can do that. And I hope at the end of my career, those two patients that I said I had no idea who was going to do worse and who was going to do better, that I can do a much better job taking care of them. So thanks so much for inviting me to speak, and I'm looking forward to the discussion.
Thank you, Dr. Mariani. We really appreciate you and always being an advocate for NephCure. So thank you so much. Next, I'd love to introduce Dr. Aliza Thompson, who is the Deputy Director of the Division of Cardiology and Nephrology, Center for Drug Evaluation and Research at the U.S. Food and Drug Administration, as what you know as the FDA. And a big part of our progress has been our relationship and working together with the FDA over the years. And Dr. Thompson is going to talk a little bit about how the FDA has a role in advancing our new therapies for our patients.
Great, thank you so much. Hopefully everyone can hear me. So just like the others, I want to thank you for including me, and thank you, Kelly, for all your hard work in putting this meeting together. As others have noted, this is a tremendously exciting time for drug development for protein-spilling kidney diseases, and we've gotten here today, I think, very much because of NephCure's efforts and the efforts of the larger community. We're here today with drug companies interested in developing treatments because you all have made that possible. So congratulations. And although there's obviously a lot of important work ahead of you, I think it is important to pause and celebrate how far you all have come. I think as noted by others, successful drug development takes a village, and patients really need to be at the center of that process. And so I want to use my time with you today to share a regulatory perspective on drug development for rare kidney diseases, and also speak a little bit about patient-focused drug development. Kelly, next slide, please. Great. As Laura alluded to, drug development for rare diseases is in many ways more challenging than drug development for common diseases. That said, I want to point out that we have had a tremendous amount of success in developing therapies for rare diseases. As seen on this slide, if you look at our drug approvals in the US in 2019, over 40% of those were for rare diseases. Next slide, please. Right. So why is that? I think one important factor here was the Orphan Drug Act of 1983. And some of you may be familiar with this, or some of you may not, but essentially what this act did was it provided for economic incentives for companies to start developing therapies for patients with rare diseases. And I just want to highlight here that the idea and that the law had nothing to do with the standards for drug approval, but it was more about making this financially attractive to do. And I think underlying this is the concept that we want to provide financial incentives, but we also want to make sure that patients with rare diseases are entitled to safe and effective therapies, just like patients with common diseases. So what does it require to get a drug approved in the US? I want to make the point that the standard for establishing effectiveness is the same for both common and rare diseases. And how we describe that standard, we call it substantial evidence of effectiveness. That said, we recognize that there are differences between rare diseases and common ones, and that the trial designs won't be the same. And I just want to highlight that the regulations therefore provide for flexibility and scientific judgment and how we go about determining whether a therapy provides substantial evidence of effectiveness. In other words, helps patients, helps treat their disease, helps give them better lives. Also want to emphasize that the fact that we have flexibility, that shouldn't be interpreted to mean that anything goes, that whatever a drug company wants to say is, should be sufficient to get a drug on the market is sufficient. As colleagues of mine have said, because of the challenges associated with developing drugs for rare diseases, how we should interpret the flexibility is really just that we need to be smarter and we need to work more closely with the patient community when we design development programs for rare diseases. Next slide, please. Great. The other thing I want you to be aware of is that FDA has a number of programs to expedite the development of therapies intended to treat serious or life-threatening diseases with unmet medical need. And I just want to point out, I list some of these on the slide. In the interest of time, I'm not going to talk about them, but in essence, what these programs do is they allow for more interactions between the drug developer and the FDA staff. They give or allow greater flexibility in the design of these development programs, and also can shorten the timeline for agency review of the marketing applications. I also want to highlight that I think all of the diseases that we're talking about today are serious or life-threatening, and certainly a number of drug development programs in this area have qualified and in the future can qualify for some of these programs. Next slide, please. Great. So where do I want to go from here? From here, I'd like to shift to the idea of patient-focused drug development. I think it goes without saying that the people living with the disease should be at the center of the drug development process. And yet, some could argue that as we develop this whole research enterprise, we lost track of that. Fortunately, we as a larger community are working to address this issue. Next slide, please. So I think some of you are familiar with FDA's efforts in the space, but in brief, the overarching goals of the initiative can be, I think, really divided into two buckets. One bucket, or one key component, is listening to patients and learning from patients about what matters to them. I think the other bucket or component is really what I'll refer to as the science aspect. It's certainly one thing to say that we want to incorporate the patient voice in drug development, but a separate question is how we effectively and systematically ensure that we do so. Just to walk through a few of these bullets on this slide and the next. So one critical issue that we need input from patients on is how to best design trials to facilitate their enrollment and really minimize the burden of participation. Again, making it easier to do these trials effectively and efficiently. Next slide, please. Other aspects about this include understanding patient preferences and your willingness to accept trade-offs between treatment benefits and risks. And finally, another key component of this is understanding the information that's most important to you related to treatment benefits, risks, and burdens, and how best to communicate that information to you to really support your decision-making about whether a therapy is right for you. Next slide, please. So fortunately, we as an agency have been able to participate in a number of patient-focused drug development meetings where we get to hear from the patients about what matters most to them. I'm really excited to point out that the NephCure Foundation has been involved in some of these efforts. And really, we as a community, as members of FDA, have been very fortunate to hear from patients and really want to emphasize how powerful the patient voice is in terms of both educating us and also reminding us of the urgency with which we must act. Next slide, please. The other thing I want to highlight goes back to some comments that Laura made. You know, at our patient-focused drug development meetings and in other venues, we've heard loud and clear from patients very much powerful stories about the impacts of the symptoms of the nephrotic syndrome on their lives. And as Laura alluded to, the community has been working on instruments that can be used in clinical trials to really assess the important symptoms and impacts so that we can develop therapies and say that the therapies improve these symptoms. And we as an agency want to support those efforts. And so we issued a funding announcement, essentially trying to solicit applications for and provide money for work in this area to support the development of instruments that could be used in clinical trials for the nephrotic syndrome. And I very much hope that this initiative and the applications that are submitted and the grants that are provided will contribute to the larger efforts undertaken by the NephCure Foundation and other groups. Next slide, please. Great. So just in closing, successful drug development takes a village, and you, the patient, really need to be at the center of this. So thank you again to the NephCure Foundation for everything you've done, and we look forward to working closely with you in the future.
Thank you so much, Dr. Thompson, for your ongoing partnership. And I just love when patients can hear the heart of the FDA. So thank you for sharing. Next, we're going to move on to the current explosion in trials and hopefully the potential new therapies that are arriving soon. And with that, I'd like to introduce Dr. Debbie Gipson, a professor of pediatrics at the University of Michigan. She directs the Kidney Research Network, and she's a principal investigator for CureGN and co-investigator for Neptune. She also designs and conducts clinical trials for children and adults with nephrotic syndrome associated kidney diseases, and has really been a cheerleader for increased, as you'll hear, for increased inclusion of pediatric patients in clinical trials. Dr. Gipson?
Thank you so much for the introduction. I appreciate it. And again, I want to add my voice to the congratulations to NephCure on this 20th anniversary. What a fantastic milestone. When we think about and reflect on the first speaker's presentation, as Melanie talked with us about the fact that when she first arrived with this disease, with nephrotic syndrome, there were a few therapies. And she's young, so we get to fast forward in just a few years and recognize this outstanding progress that has been made and that continues to be made in large part because of the advocacy of patients like Melanie, their families, and organizations like NephCure. So well done, all of you. When we think back even just 5 years, though, we don't even have to go back to 20 years of NephCure, but we remember that there's a lot of preparatory work that you begin to hear about over the last few speakers. And up to 2015, that work has driven us to look at essentially a snapshot of clinical trial activity. And what you see here is that activity that's demonstrated by disease example and by phase of clinical trial to help you know how many clinical trials were available to patients to participate in trying to drive our options for therapies forward. And then you move forward into 2020, and what you see is, again, this amazing change in just the number of markers that you see here, again, across all diseases and across all phases. And supported and generated by multiple sponsors, what we're seeing is a lot of activity all the way from preclinical, before we're ready to bring the therapy to humans, to early phase, let's make sure that we're thinking about safety in humans, even who don't have disease, to middle and end phase where we're beginning to think about these therapies that can move us along so that we can change from we wish we had something better to we have clinical trials, hang tight, to what we do believe is in the very near future an ability to change the way we prescribe and treat effectively and safely our individuals in our population who are affected by these kidney diseases. Go ahead and move forward. So what stimulates drug development activities, and you've heard some of these already. So we think about the discovery in the disease mechanism. If you're trying to do a better job of treating a patient's disease, then you need to understand what that disease is. And historically, we've done a great job of thinking about diseases in bag categories, labels, but not so much from a scientific perspective of bringing us down to the real scientific cause of the disease and what's driving the disease to begin and to continue. So there's been a lot of work really stimulated by investments from NephCure, by investments from the National Institutes of Health that have helped to bring science forward for us to understand better disease mechanisms. The FDA partnership, which you've just heard about, really defining our options for drug development in these rare diseases, working with us as a whole community to think about how we can identify when an individual is doing better, right? Which is our signal for a therapy maybe going in the right direction. And that took some really good leadership and some change that we've been experiencing over the course of the last many years. We needed, as you've heard, better outcomes for clinical trials, better outcome measures, and those, again, are resting on some of the studies that have been generated and developed with patients, for patients, using resources made available by NIH, by FDA, by industry partners, and by foundations such as NephCure to really help us to do a better job of saying, does this outcome that's closer to the patient's beginning of their disease, right, or the middle stage of their disease, does that outcome help us to be able to reliably predict what will happen in the later stage? And will it change when we try a new therapy? Will it get better, and will that get better translate into a signal for us to be able to think that a patient is improving and maybe changing off the pathway of disease progression or disease activity? You're doing great. And so we think about then how we move on to drug development, and we think about two major pathways. We're going to think about drug repurposing and novel drug development. Drug repurposing is really an example of a therapy that's been developed for another disease. We're going to call that disease 1. And then what happens is, because of observation of a patient who had disease 1 who used that particular therapy and look at also help their kidney disease, right? And someone looked up and said, that makes me think that this may be helpful to other individuals. Sometimes it's because a matching of a mechanism of a therapy to a mechanism that we think is important in kidney disease. And all of a sudden what happens is all of the hard work of drugs that were developed for a different disease can be repurposed and reconsidered for the use in patients who have diseases that fall into our nephrotic syndrome umbrella. And our off-label examples of drugs that have been repurposed that we really use in standard of care today include therapies like cyclosporin and tacrolimus, mycophenolate, and rituximab that are used in these diseases. And while we say thank you for those, we ask for a little bit more, as you've heard, a little bit more precision and effectiveness. Go on to the next slide. And so it brings us to essentially moving towards drug development that is anchoring off of the science that has been in part highlighted here today, and also that is well beyond the scope of 10-minute presentations, that begin to think about patients not by their disease scarring pattern on microscope kidney biopsy, but in the details of their disease mechanism that we might find in more subtle markers of disease. And so we think back about essentially some of the information that's been generated in a more precise, patient-specific, and then patient subgroup-specific way to ask the questions, how are patients more similar by disease mechanism? And that first panel shows essentially the green-red spectrum of transcriptomics from kidney tissue. moving, essentially identifying certain subgroups that may then hang together because of a disease mechanism that may be quite relevant to that patient subgroup. And then what happens is we say, so now prove it, did that subgroup matter? And so then we ask the question, did the patient's health outcome, ability to control proteinuria, ability to maintain or preserve kidney function, did that essentially, those health outcomes align with the disease mechanisms? And so we, again, look at that longer term view from patients who've been part of our long-term observational studies. And when mechanism seems to match outcome, we look up and go, aha, we have this opportunity now to think about drug development in that matching activity. And then we find a way then to build essentially therapy, clinical trials to test therapies for drug development. An example of this one, again, is a trial that's open today that really rests upon this type of scientific discovery that is doing its job to be able to identify patients who have signals that a particular disease mechanism is relevant for that patient at that time. And then it selects a therapy that actually may be well matched to that mechanism to ask if the signals look like they're aligned and if we could make it better in a relatively short period of time. Next slide. So that allows us, actually, that first example is actually a repurposing drug example, but we use the same techniques, the same strategies to be able to think about novel drug development. So again, we go back to the patients and we think about patients by disease mechanism, by disease severity, the beginning disease, the midpoint of disease where perhaps they've not responded to the most common therapy, first therapy, for example, or in a native kidney population, meaning the kidneys you're born with, or a transplanted kidney population trying to prevent recurrence of disease. We think about now that big spectrum of patient population, right? And we think, what is the disease mechanism in play? And then in that disease mechanism, if we don't have a therapy that we can repurpose and reuse for that mechanism, we begin to work on novel drug development. And many of the trials that are available today that were listed and referred to in the 2020 summary include novel drug development that have been generated using these types of techniques, which is incredibly promising and really a true credit to NephCure's investment and progress. Next slide. And so finally, and as Kelly reminded you, I'm a pediatric nephrologist, but I manage trials for adults and children. These are really important. But we remember that these rare diseases of nephrotic syndrome affect children and adults. And so we ask ourselves the question, in that whole spectrum of disease, when are the diseases the same, right? When are the outcomes the same? And when are the needs for therapies the same, such that this should drive the inclusion of children in drug development so that we are generating solutions for therapies, solutions through therapies that were developed for the entire spectrum of the affected patient population. So this becomes important. And again, part of our collaborative work with the FDA has helped to define better and better pathways for the inclusion of children safely into clinical trials, and so that children can be part of their own solution and part of the beneficiaries, to be perfectly honest with you, of drug development in their lifetime and in their generation, as opposed to always waiting for the next generation. That really ends what I wanted to say today, except to say really congratulations. I'm just delighted to be a little bit of the anchor for this particular panel, but also to say the hard work that has been done over the past 20 years through NephCure and through the really amazing volunteerism and partnership with all the patients and families is really bearing fruit. And so it's fantastic to be a little part even of this activity.
Thank you so much, Dr. Gipson. We really appreciate you. And I'm going to change to our gallery view now. If our presenters want to turn on your videos, and we're going to take a few questions. So if you have any questions, feel free to type those down. I see that. Thank you to our panelists for answering a lot of the questions that have been coming in already. But if anybody has any current questions, feel free to type them in. I do see one here that I'm going to read. And it says, my kidney biopsy report say very weak granular positivity for the PLA2R along capillary loops with IgG and C3 complement. PLA2R serum titer is negative. I also have a history of taking herbal medications with metal content in it. Is there a chance of secondary membranous nephropathy? That's a doozy, the first one. Anyone want to take a stab at that?
I don't think we know the answer to that. It is clear that membranous went from being an idiopathic disorder to a primarily autoimmune disorder. As we have seen, there's autoantibody against different targets. But what really triggered autoimmune response with the exposure to herbal remedies, to heavy metals, to viral particles is the trigger of the formation of this autoantibody, as far as it remains to be studied.
Great, thank you so much.
I would also add to what Dr. Fornoni is saying, that PLAR2 is not the only antigen that we know in membranous nephropathy now, and part of this autoimmune spectrum. So there are others, thrombospondin 7, NAL1, etc., that could be considered.
Great, thank you. Another question from an anonymous attendee. How are the racial and ethnic disparities in kidney disease being addressed in clinical trial recruitment?
That's a great question, and I'm going to grab that one if I may. The nicest part about clinical trials today is that they have a true intention and actually success in encouraging patients who are affected by kidney diseases to participate in those trials. And so what we found is this amazing generosity of patients across socioeconomic spectrum, across racial and ethnic, what I can say inclusion as opposed to divides, that has really borne out in our patient participation, which is fantastic. We do a few things though, and it's important to do these. And one is that we intentionally include participating sites that really represent an enrichment strategy for where patients live in their communities, that have that, that diversity incorporated, and that becomes important. The second is that, you know, sometimes being in a clinical trial that's not in your hometown is hard. And we do try and are quite successful actually at having resources available to help offset the travel costs for patients and their family members and a family member to actually go to wherever the clinical trial is being conducted if it doesn't happen to be in their hometown. It's really important, and it's actually important, please, for patients who are thinking about doing this, don't let those finances be your barrier. Remember to ask if a travel program is part of a clinical trial, because it's oftentimes present and it's intended to allow those doors to be wide open. And it's just really, it is so important to be able to participate. There are clinical trials that are open and that are being developed that are very specifically addressing disease mechanism that may anchor within a subgroup by race. And it really is not really race, it's actually a genetic particular component that, that we tend to sort of we consider as co-locate with race, but that becomes really important. It's a disease mechanism related study that's a genetic finding that helps a person to get in. And so again, tailoring the mechanism with the therapy option in the clinical trial is incredibly important. And so it's really fun to actually see the trials being developed that are right fit for the patient community.
Kelly, I know you need to move on, but if I could just add one aspect to this as well. We at the FDA are very interested in this issue. We need our trials to be done in the people who are going to use these therapies. And so we have put out guidance related to best practices. And I think a lot of this is about getting and making sure that when we design the trials, we design them in the way that they're not overly burdensome, that it makes it possible for people who maybe are socioeconomically disadvantaged in various ways. It recognizes the challenges but really makes it possible for them to participate. So just want to echo that this is a really important issue and one that we, I think, as a community are working to address.
And I want to add to that, even the companies that you are actually developing drugs, understand the importance of this, and at the moment they are investing in community outreach program to make sure that the most novel therapeutic options are also offered to minorities and to the communities. So I think that's a very important question.
Thank you so much. That is a very important question indeed. So the next question is, how do you as doctors empower your patients to increase self-management of their disease?
Maybe I'll take that one. I think as an adult provider, I just feel like the most important thing is having my patients be my partner. So, you know, I think a few things. Number one, every new patient that I meet, I show them the NephCure website. I show them the Kidney Health Gateway so that they know how to follow along with exciting exciting developments in trials and things like that. I want my patients to come and ask me questions and to say, hey, I heard about this thing, what do you think? So, so connecting them with resources that are, in my opinion, more reliable, I think is really important. And then secondly, you know, I hope my patients become nephrologists. So they know how to open, it's my favorite thing when I get an email from my patient like, hey, I saw my labs already. I think we should do this on my tacrolimus. I'm like, great, yes, you're exactly right. So it's a lot of education about every single drug, every single lab test. So they can follow along and eventually start to tell me what they think they should do next. Because truthfully, every patient's different and they become experts in their own disease.
Okay, and I think we have time for maybe one more. Thank you so much, Dr. Mariani. Many doctors prescribe so many different cocktails for these diseases. How do we know as patients which ones really are better than others? I think this is a great one to end on.
So many of us actually have an answer to that question. So, you know, we'll make them all short and see if we can do a round robin. How about that? You know, the cocktails for therapy for these diseases have been really generated from a true desire by physicians and patients for progress and improvement, right? And what's happened is that in the midst of that sort of drive and desire, people have done their best. And oftentimes then these regimens or combinations of things are not necessarily go through the type of rigorous testing that we ask of our clinical trials, to be perfectly honest with you. We do our best. And it reminds us to sort of stop and say, you know, in a patient care one by one, we start a therapy, or we start a new therapy, and we ask of that patient, are you better? Is your disease better? How are you feeling? Are you tolerating this therapy? Are we making progress? And that sometimes feels like the best that we can do. But actually collectively, we can do much better. And that collective action means to be able to say, you know, where is that trial, study, participation that we as clinicians and that patients individually can look up and say, I want to be part of that. I want to donate my data, my information, so that people can analyze that as a group and look for patterns of improvement and begin to build studies that really are relevant to me as a patient and as, you know, me as a representative of a patient community. And so be part of that. That kind of question is incredibly important, not just at this panel, but it's incredibly important when you're going to your physician, just like Dr. Mariani, Dr. Fornoni, Dr. Shankland, for all of us to sort of look up and say, great question. Let's talk about what we know, but let's also talk about what we don't know. And let's talk about what the opportunities are for you to help us to be wiser. And it becomes incredibly important to do. Passing the baton now.
I want to second that. No, it's only through being able to analyze and study more patients that volunteer to donate samples to research that we can eventually, hopefully, we will be able to take personalized decisions on which drugs would benefit better patient versus the other, just like we have seen our colleagues in cancer therapeutics do now for a couple of years. I think we have the capability to do this in the kidney space as we collect data from natural history studies that NephCure International Regulatory Agency, the NIH, have been so instrumental in supporting throughout the year.
I would agree with what has been said by Dr. Gipson and Fornoni in terms of cocktails. I think that the field is changing dramatically. I think that it's becoming evidence-based. In other words, as has been said by Dr. Gipson and Fornoni, what is in the literature and how do you as a patient fit into this? And so historically, this has not been done as well as it is today. And I think this is the change. And yes, we ultimately want to go to a precision medicine model for individual patients. I do think we're close. We're getting close. I think, for example, membranous nephropathy is in the glomerular podocyte world is probably the closest to date. And I see, you know, cocktails, hopefully not just being used based on old data, but based on exciting new data, as Dr. Mariani explained, some of these beautiful studies that are coming out with these consortia that are really going to take population studies and narrow it down to individual patients. We're getting closer.
Yeah, and I think I would just encourage you to, you know, never feel that you can't ask your doctor, you know, where they come up with that idea. So, you know, have you tried that before? Have you seen that work? You know, is it something that we're borrowing from another disease? And also, you know, if you ever feel like you're just feeling more sick than better, you know, I'm never offended if someone wants to have a second opinion. That's fine. Like, you know, so I would just encourage you to be your own advocate, and you know, every doctor should be fine with that.
Well, thank you. I want to end by thanking all of our panelists and helping us celebrate our 20th anniversary. It really is emotional listening to everyone talk about the progression over the last 20 years, but also it's exciting looking forward to the following 20 years. So thank you so much. Those of you who are attending, I just want to let you know that we will be emailing out the recording and the slide deck for this presentation so that you can go back and re-listen and look at the slides. I also want to encourage all of you to join us as we continue to celebrate our 20th anniversary. We have events coming up. You can follow us on our social media. Please visit our website under events and consider joining us for our 20th anniversary countdown to a cure virtual gala, where we'll be highlighting even more people, events, moments over the past 20 years. And we would just love to have you join us. Again, thank you, panelists, and everyone have a great rest of your day.
Thank you so much.
Thank you.